Good morning, class 10th. Children, last time when I took your class, we discussed about the structures of arteries and veins under the heading blood vessels. And today we will be moving further with capillaries. Right? So, in our body, three types of blood vessels are there. Depending upon slight structural and functional differences, they are arteries, veins and capillaries. We will be discussing the diagram again, but as of now just undergo the important features of capillaries. These are the finest thread like branches of arteries which are capable of reaching each and every cell of your body, though they do not enter the cell, rather they communicate with the cells with the help of tissue fluid. We will be discussing this slightly later on, but as of now you just understand that these are the profused finest thread like branchings in your body emerging out of the arteriole, and they are basically meant for the purpose of supplying blood, nutrients, oxygen, everything to each and every cell of your body. The first characteristic feature of a capillary is that it is made up of, it is not very thick, it is not having 3 layers or 2 layers, it is made up of only a single layer of epithelial cell, that too the squamous ones, the flat ones. Right. Second thing is the capillaries are capable of vasoconstriction and vasodilation. Children, you need to understand vasoconstriction. Vaso term is the prefix used for blood vessel, and constriction means shrinking or getting narrower. Right. Similarly, dilation means expansion. Right. So, shrinking of the blood vessel or the expansion of the blood vessel, this is the peculiar feature of a capillary. Depending upon your body requirement, this capillary may undergo vasoconstriction or vasodilation. Now let us discuss under what circumstances this occurs. When you move, when you are standing under the sun for a long time, suppose under the scorching heat, your face turns red, the hot flashes, the redness on your cheeks, it is there. Right. Why? Because this is something where the body tends to lose the heat as maximum as it can, and this is done by vasodilation. Vasodilation is done under the condition of scorching heat. Why? Because when these capillaries dilate, they allow maximum movement of blood, maximum circulation of blood beneath the skin, and this maximum circulation is basically for the purpose of loss of heat. Right, but since it is beneath the skin, much blood is flowing, and this is something that turns your face red. Right. Similarly, vasodilation occurs just opposite of vasoconstriction. So this is the shrinking or narrowing of a capillary, and this occurs in chilling conditions. Suppose when you dip your hand in freezing water for a long time, or you are standing in the snow for a longer time, or you are feeling much colder, it's shivering cold and you are feeling very cold, at that time if you have ever noticed your face turns blue. If not face, at least you must have noticed the tips of your fingers and your lips, they turn blue. Why? Because these are the areas, particularly the lips, these are the areas where blood capillaries are very, very superficial, and any change in the blood capillary would be reflecting here. So, when you feel cold, at that time vasoconstriction occurs, because your body needs much of the heat to be restored inside, and in that case, it cannot allow much blood to flow through the capillaries. That's why the capillaries constrict to allow, to restrict the movement of much blood beneath the skin, and this is something, since very less blood is flowing through the capillaries, your face turns blue or your lips turn blue. Right, children? So capillaries, they are actually very effective. They help you to combat excessive heat or extreme cold. The third thing here is the main function of capillary, that's to allow diffusion. Always remember, a capillary does not get into the cell, it actually is allowing diapedesis. I hope you people still remember, diapedesis is the oozing out of blood plasma along with some leukocytes particularly the lymphocytes, across the wall of the capillary. And this is possible just because the lymphocytes, since they are WBCs, they are amoeboid in nature, so they can pass through a very minute pore very easily, right? So the communication between the blood flowing in the capillary and the cells of the body, this communication is just maintained by filling up the tissue spaces with that fluid that you call tissue fluid. We have already discussed what is tissue fluid, right? Another one here, what can capillary pass through? So whatever is present in the blood plasma, that can be passed through to the cells. You may list it up as the exchange of oxygen and carbon dioxide, exchange of passing on supply of nutrients, antibodies, hormones, any nutrient that's present, and also to eliminate the waste in form of carbon dioxide and urea. This is also exchanged. So exchange of substances, you may say, through diffusion, simple diffusion, right? And the last one is allows diapedesis. I have already told you what is diapedesis. It is the oozing out of blood plasma. That means apart from RBCs and platelets, only the liquid content of blood mixed with salts and say hormones, and apart from this, the white blood cell, particularly lymphocyte, this oozes out of the capillary wall, being amoeboid in nature, right, children? So these are the peculiar features. They have included the functions too. This is everything that you need to know about a capillary. And if you can see that I have also drawn a diagram here, again we are doing this is just to let you know that if you take out a slice, now I was telling you that day I couldn't explain you well. Suppose if this is a tubular pipeline and then you have to see what is there inside, you have to take out, you have to slice it off transversely, right? So this is what you have done here. Suppose this is an artery, you have transversely cut it into a section. This is how it will be appearing. So artery, you can see the three layers, very distinct layers, light red, dark red, and then dotted red. And these three layers are tunica externa, tunica media, tunica interna. The innermost layer goes as the wall of lumen is epithelium, endothelium rather, and the lumen of the artery is very, very narrow. You can see that. On the contrary, when you notice vein, the three layers are still there, but they are very thin. They are not very thick. That's why the vein is not very much elastic, and that's why the lumen is very, very wide. So arteries can put much pressure. They can exert much pressure over the blood flowing within. Why? Because they have thick elastic walls that can help in exerting higher pressure, which is not the case with veins. And the last one here is capillary. You can see the single epithelial layer is there, and this is how you recognize if the straightaway cellular composition is shown and no layers are there, muscular layers are not there. This is a capillary. Remember this diagram because you have figure based questions on this diagram, right, children? So I hope this much is clear to you. I'll be clearing one more thing along with this, hepatic portal vein. Now this is something you have to listen to me very, very carefully about this, children. If you can see, I have drawn two diagrams here. First of all, you come to the normal diagram, right? Just hold on. Let me select a pen. Orange. Fine. So now you understand that under normal conditions, when you talk about the flow of blood in arteries and veins and capillaries, what is the course of flow? You see that this thicker most area, this is the artery, right? You see that it is branching into two. That is, I selected a thinner one, sorry. If you see that it is branched into two, these are the arterioles. Then further, the fine thread-like branchings, you call them capillaries, right? You must be wondering about why I have drawn half of the capillary red and half blue. It's just to tell you that when this capillary is involved in supplying oxygenated blood, I have drawn it red, and when these capillaries are involved in recollecting the carbon dioxide mixed with blood, I have drawn them blue. So it is as simple as that. And surrounding will be the tissue fluid, which is filled in the intercellular spaces around the cells. I have drawn this diagram earlier too, so you might be finding it very, very familiar. Right, children? So after doing the work, capillaries reunite to form the vein, right, and then venule, and then the vein. This is a normal course of flow, that an artery divides into arteriole, and then arteriole further divides into capillaries, and capillaries reunite to form the venule, and then vein. So artery, capillaries, vein, this is the course of flow. You have to keep one thing in mind. If you can see, I have drawn this dotted outline just to show that this is an order within any organ. If you talk about any organ of your body, this should be the course of flow, that the blood vessel entering the organ should be an artery, because it is having the purpose of supplying oxygenated blood, and the blood vessel which is exiting out should be a vein necessarily, because it has the purpose of collecting deoxygenated blood back to the heart, right? So entry of a blood vessel should always be with the artery, and the exit should be with the vein, because you have to supply oxygenated blood to an organ, not the deoxygenated one. You have to eliminate or remove the deoxygenated blood from the organ. So this is the normal course of flow. I hope you people have understood. Artery, capillary, and vein, this is the normal course of flow, right? But now you have to compare. There are certain exceptions in your body which are very, very important to be understood here, children. Just come slightly above. Hold on. Now you just see this upper structure, this one, the above one. I hope you'll be able to see. Hold on. I'm talking about this one there, the above, hepatic portal vein, right? What is the difference here? If you can see, I have drawn two diagrams here. On the right side it's intestine, and on the left side it's let me fix it here, and I need a pen. Fine. So on the right side is an intestine, and on the left side I have drawn a liver, right? I have drawn these two organs because this is the case where hepatic portal, the portal vein system is there. What you have to see, generally the logic says that in an organ, the entering blood vessel should necessarily be an artery, and the blood vessel that is exiting the organ should be a vein. Fine. So here in intestine you see that the artery is entering. So the artery which is entering the intestine is an intestinal artery, right? You have to see this, that the main artery is aorta, and then depending upon in which organ it has to supply blood, it gets branched, right? If it has to enter the intestine, aorta is a big artery, it cannot enter straight away into small organs, so it has to branch further. So intestinal artery, if the artery has to be branched towards the intestine, it is renal artery branching off from aorta if it has to enter kidney. So here I have drawn an artery which is entering intestine. You will be writing intestinal artery. You can see that incoming is red, that means oxygenated blood. Branching up to the level of capillaries, that means oxygen supplied and CO2 collected. Fine. So now when this artery will be, when this vessel will be exiting intestine, it should be a vein. Right, we will be labeling it as a vein, but what is important here, that intestine is the site of nutrient absorption. That means at this place blood is equipped with, it is loaded with nutrients, and these nutrients are supposed to be stored somewhere or processed somewhere so that they can be assimilated in the body. Right. So the blood vessel which is exiting intestine is having carbon dioxide but also loaded with nutrients. You have to fix those nutrients up. So it straight away goes to liver, because liver is an organ which is basically meant for fixation. Right. So it will enter the liver, but This is a vein. You can see it's blue in color. Exiting intestine is necessarily a vein, right? But it has again entered the liver. How come a vein can enter an organ? An artery should enter an organ, but here, because of the purpose of fixation of nutrients, this vein re-enters the liver, rebranches up to the level of capillaries, so that glucose present in the blood can be fixed in the form of glycogen, so that the amino acids present in the blood can be deaminated and urea is produced, so that the toxic waste ingested through food can also be detoxified. So liver does everything, whatever processing is needed with the food, and then the blood vessel exiting the liver is hepatic vein. I have already told you that hepatic term is used for liver, right? But the only difference is, since here the capillary level, this is a blood vessel, this particular portion, right, exiting intestine and entering liver, this portion is beginning with the capillary and ending up in the capillary, right? So you will be calling it hepatic portal vein, right? Generally such arrangements are portal vein system. You call any blood vessel that starts with the capillary, exits out the organ, then enters an organ, again end up in capillary, you call such systems as portal systems. But since we are specifying here the movement of blood between intestine and liver, that's why we are calling it hepatic portal vein. I hope this much is clear to you, children, and if you have any queries regarding this much of portion, please let me know, right? I'm concluding this video here and I'll be proceeding further with the next video.